very much. My name is Susanna Wilkie. I'm the CEO of Neurotrope. I will give a short summary of the science and the opportunity in our corporate progress, and I will hand it over to Dan Alcon, who also gave the keynote lecture today. I just want to mention that we're very happy to have Dan here today, and the importance of his life's work. He has been working on this science and establishing a comprehensive mechanistic framework for synaptic regeneration over the last 45 years, and worked for the NIH for 30 years and at the Planchette Rockefeller Institute for 15 years. And the science has attracted over about $300 million of funding in total, $200 million in grants and $60 million in direct investment in Neurotrope, and grant money that is representative in the Bryostatin that was donated by the NCI to Neurotrope, adding up to almost $300 million. And the significance of this is that tens of millions of this money was spent in using electron microscopy, serial electron microscopic reconstruction work, to meticulously and diligently look at what happens initially on learning and then on neurodegenerative diseases. So Dr. Alcon was able to establish PKC epsilon as a key mediator in associative learning. And then he turned around and he says, well, what is the role of PKC epsilon in the generation of diseases? And he found that on lower concentration of PKC epsilon, we actually see different manifestation of neurodegenerative diseases and neuropsychiatric diseases like stroke, TBI, traumatic brain injury, Fragile X, for example, as a neurodevelopmental disease, depression, and other areas, Parkinson's. So he found that the central enzyme PKC epsilon is critical first in associative memory, but also in formation of neurodegenerative diseases. We believe that so we are a little bit out of the box, as you can tell. And the part of the reason is that Dr. Alcon initially didn't start out to treat Alzheimer's disease. He was interested in, as I said, associative memory, and so understanding the connection between loss of memory being tightly correlated with loss of synapses and synaptic networks. And we believe if we can regenerate these synaptic networks, that we actually can reverse cognitive decline in Alzheimer's disease. So our clinical program is using Bryostatin, which is not a statin, it's a marine natural product, to activate our master enzymes, PKC epsilon, which Dr. Alcon identified as a master switch, to regenerate and revitalize neurons, and with that, revitalize and regenerate synapses and synaptic networks. And we'll believe that this is the regeneration of the synaptic networks that ultimately gives us clinical efficacy. So we are looking for a large impact in clinical trials. And the reason for that is that every neuron has about 100,000 to 200,000 synapses. And if you can re-stimulate these synapses, you will regenerate the wiring that connects to different memory pieces to give you a better cognitive profile. With that, I want to hand over to Dr. Alcon to give us more detail. We are phase 2 at the moment. We are ahead of a critical, pivotal inflection point in the development of our company, which is the release of the top-line phase 2 data in severe Alzheimer's patients end of April. And this is to say that we believe that even at this very advanced stage of Alzheimer's, we can regenerate synapses and synaptic networks through the activation of PKC epsilon with Bryostatin. Another critical company inflection point actually was happening today. We announced the uplisting to NASDAQ, which shows the progression of our company onto the major trading exchange. We also really have had a number of major corporate events. One of them, just to highlight, is the licensing of the accelerated synthesis of Bryostatin, which has been viewed in general as impossible. It's a large macrolide with 11 chiral centers, and we now have a commercially very attractive pathway to actually get to the commercial quantities that we need if this is successful. And again, the next corporate event is the release of our top-line data, where we believe we can hopefully show reversal of disease versus just slowing down progression of the disease. So again, with that, I hand it over to Dr. Orphan.
So can everyone hear me from this perspective? What I want to do is just briefly recapitulate what I talked about a couple of hours ago in my plenary talk, rather than repeat all the details. Then maybe we can have an opportunity to have a discussion and question and answer period. Why this target is closely linked to synapses that are lost in Alzheimer's disease has to do with looking at autopsies. It's not only our studies, but a whole number of other studies that has shown that there is not only a deficit of PKC epsilon in the Alzheimer brains of freshly frozen samples as compared to control brains, but other laboratories have shown that there is a loss and deficit of BDNF and other growth factors also in the Alzheimer brains. So that pathway, its absence and its deficit, is reasonably linked to the loss of the synapses that are so characteristic of this disease. Remember that I mentioned that early on in the Alzheimer's disease autopsies, what you see is loss of synapses, often in the absence of amyloid plaques or tau. In addition, if you look at the transgenic mouse models, particularly those that track what human pathophysiology shows, you see the first thing you see is the PKC epsilon deficits, very similar to what we saw in the Alzheimer brain. Samples from Harvard, you see the loss of synapses, you see the elevation of soluble A beta oligomers, and if you treat with Bryostatin in the transgenic model, you rectify that. You not only increase the activation of PKC epsilon, but you increase the synthesis of it. So there's a genetic target there that allows the drug to actually increase the synthesis of the target PKC epsilon, which, as I mentioned before, is related to human disease in that ApoE3 normally regulates the synthesis of PKC epsilon, whereas ApoE4 is blocked from doing that. Those kinds of studies suggest, before we did any studies in transgenic models of great length or clinical trials, that if we rectified those abnormalities, we would rectify the cognitive deficits as well. And it suggests also that, given that in autopsy brains, when you actually correlate the loss of synapses with the cognitive deficits, they're closely correlated, whereas with amyloid and tau, they're not closely correlated with the cognitive deficits. That is not to say that the amyloid and tau are not important. We believe they are. And we're fortunate in the sense that Bryostatin and other members of our platform, many of which are new chemical entities, are actually multimodal in their efficacy. They do have the apoptotic neuroregenerative quality to them, rejuvenating neurons that are dying. They do have the synaptogenic quality to them. But in addition, they directly address all the major enzymatic pathway critical points for regulating the generation of A beta oligomers, actually preventing it, and regulating the hyperphosphorylation of tau and preventing that. So in that sense, it's very well positioned to fulfill multimodal efficacies. So I'll just show a few slides. Let me just see if I can do that with this. So this is the drug which you saw before. It was formerly isolated from 18 tons of natural material by the NCI to get about 10 grams of it. And that's why we're so pleased that we now have a exclusive synthesis licensed from Stanford University under Paul Wender's inventorship to Neurotrope for all neurologic and psychiatric indications. And this is an illustration of what happens in normal animals. This is another thing that we've tried to emphasize, using endogenous signals that are normally present in the animal brains and the human brain, and then bring in exogenous signals, then bring in drugs. But first tap into the endogenous signaling pathways. This is a spatial maze, This is a spatial maze paradigm, and as the curve goes down, that's a normal curve of spatial acquisition. The black is when you have, in some way, an interruption. This is retention curve over here. You can see that normally you have an increase in the retention of the target focus in the target quadrant, even after the animal no longer has an underwater platform. And when you treat with Bryostatin, you tremendously enhance that. That's in normal animal models. And I think that's another distinguishing feature of the way we have gone after this. We looked at endogenous normal learning and memory pathways. None of the other pathways that are currently being accessed by Namenda, by Aricept, Tacrin, galantamine actually enhance normal learning and memory in this way, activating normal signaling pathways. They act on specific parts of it, but they don't integrate and actually enhance learning and memory as a whole. And then along with the acquisition, you can count the mature synapses with electron microscopic reconstruction. And you can actually show that there is an enhancement in these mushroom spine synapses. Up in the right-hand corner, you see what a mushroom spine synapse looks like. And you can quantify those. And they not only have macroscopic, they have microscopic features that identify them, and you can count them. And to me, it's quite amazing that in a normal hippocampus, you train an animal and you increase. You very significantly increase in the lower bar graphs. With just normal learning and memory, you enhance the number of mature spine synapses, and then with Bryostatin, you enhance them still further. And then, as I mentioned before, that there are multiple reasons why we have implicated this particular target. This is important because this illustrates that it gets to the blood, to the brain, over across the blood-brain barrier, so we can inject by a single IV infusion into a mammalian model. Then we can measure the PKC activation and the quantity of the PKC in the brain as a function of the injection. That way, in our phase 2A trials, which we've just completed and will be published in the next couple of weeks, we can actually see the Bryostatin going up in the human patient. Then you can see the PKC epsilon going up to a level in the blood that we can then extrapolate based on these experiments exactly what's happening in the brain. Although we're not measuring directly in the brain, we can extrapolate what's happening across the blood-brain barrier. What's amazing is that when you do in vitro experiments, so for example using cultured human neurons and networks, you can define the dose response efficacy range is about 0.2 to 1 nanomole. So we're talking about high picomole range, tenth of a billionth of a mole. It's an extraordinarily potent drug. And we can show that it's getting to that range in the blood level that should correspond to what happens in the brain. This just shows what I had showed before, that when you quantify that the synapses, the number of mature synapses on the right go down, and all the way on the right more dramatically, you can totally restore that with treatment of Bryostatin or with other members of our platform. We have other drugs that we've identified and actually synthesized as new chemical entities, some of which are even more potent than Bryostatin. And this just is an example of how, as I showed before, that the learning is totally impaired. We call it a flat line. You totally restore that. This is a stroke or a transgenic Alzheimer model or a traumatic brain injury model or a Fragile X. They all show the same consequence, not the same etiology, but the same consequence of the disease. And this is a kind of synthesis looking at PSD95, which is a target of the NGF, BDNF, PKC epsilon cascade, and you can actually measure this enhancement with Bryostatin, and you can measure, along with it, the electrophysiologic correlates of synaptic function that has been enhanced. This is another laboratory just to show that if you do it a totally different way, this was a double transgenic where they overexpressed PKC epsilon, and notice that when they overexpressed it, they completely prevented amyloid plaque. So just a confirmation with another technology. And this shows you that when you go out in time with a transgenic animal, you can eventually get to the point where you have amyloid plaques, but you can virtually prevent that with Bryostatin. And Fragile X is showing the same thing. You can see that quantified in the last lower quadrant, that you restore the normal synapse structures. This is to me, a very difficult thing to do because you have very advanced disruption and degeneration of the synapses in both the children and in the models of the disease. We not only repeated this with older and younger animals, but Fragile X Foundation did this with their model in Chile, and they saw not only rectification of the cognitive parameters, but also autistic spectrum disorders. And we'll just stop there, and I think this would be a great time to have a question and answer. You're welcome to ask what might be interesting to you.